In this video, I'm going to go through the biosynthesis of vitamin A. So vitamin A, which most people probably think is associated with things like carrots and stuff like that. Uh, so you find this in quite a few plants. I think it's actually found more in green leafy vegetables like spinach and so forth. And so there are steps in this process that we'll get to a little bit later on in this video that humans cannot do. And so that's why this is an essential vitamin, essential. We need to get it from our diet. But just to start off, so there are two places to start. And so what happens is the precursors, the very first precursors of vitamin A synthesis are these things, which are pyruvate and glyceraldehyde 3-phosphate, which these come from sugar. So once we get in a later video, we'll talk about the glycolysis pathway, and we'll go into painstaking detail of the glycolysis pathway. But these precursors come from sugar, and so does this acetyl-CoA. Uh, so both pathways start from sugar. So acetyl-CoA is actually made from pyruvate, which we will see in a later video, as I said, when I go through glycolysis. So this mevalinate pathway, which I'm not going to go into in too much detail because this is the pathway that's for making cholesterol as well. And so I will save this for for a later video when we talk about cholesterol. For instance, this HMGR, which is HMG-CoA reductase. If you take statins for cholesterol, this is the enzyme that it is affecting. This is the enzyme it's inhibiting. And so I'm not going to go too much into this mevalinate pathway, so just to quickly go over it, we start with the acetyl-CoA, which is, like I said, it comes from pyruvate, which comes from the glycolysis pathway. Through enzymatic reactions, acetyl-CoA thiolase, we generate this 2-ketone species here. Then through the HMG-CoA synthase, we add another carboxylic acid group onto it. And this double bond here, this double bond oxygen becomes a hydroxyl group. Through the reductase, we remove this acetyl-CoA and turn this into another hydroxyl group. And then, so this shows that there are two different pathways that archaea take. So humans are eukaryotic. Archaea are, so there's eukaryotic, there's prokaryotic, which are bacteria, and then there are archaea, which are something kind of in between. I'm not going to focus on that here. So then we go through the mevalinate 5 kinase, which kinase, as I pointed out in a previous video, is adding a phosphate group onto something. And so we have that phosphate added. So then we have the phosphomevalinate kinase, which adds yet another phosphate onto it. And so you will see this double phosphate named in different ways. Sometimes it will be called diphosphate, which in things like adenosine diphosphate. You'll see it like that. But you will also sometimes see it written as pyrophosphate. So pyrophosphate. So that's what you call two phosphates bound together when they're not bound to anything else. And so sometimes you will see it written as the molecule pyrophosphate rather than diphosphate. So in this case, mevalinate pyrophosphate. And then we go through the mevalinate pyrophosphate decarboxylase, which turns it into this isopentenyl pyrophosphate, which then through this isopentenyl pyrophosphate isomerase can be turned into dimethylallyl pyrophosphate. And what we see here is the carbons that this double bond occupies are changing between these isomers of each other, and that's why the enzyme that converts them is called isomerase. And so just to sort of briefly go through this mevalinate pyrophosphate decarboxylase, we see this hydroxyl group here, which we see over here on this, is going to attack this phosphate here, which is on, so this is ADP, but then it has this other phosphate here, which makes 3, so it's ATP. And so it takes a phosphate off of the ATP, puts it onto this hydroxyl here, and then the phosphate comes off, which is a reaction that is favored, and this is able to generate this positive here on a carbon, and so that gives us a place where we want to add electrons to it, because carbon ions are very unstable, unlike oxygen ions, which are a little bit more stable, or at least oxygen bound to something else like a phosphate. A carbon ion like this is very unstable, and so the electrons from this oxygen are going to come down here to make another double bond to this carbon, which is then going to break this bond as it forms double bond to this charged carbon. And that's how we get our isopentenyl diphosphate. See, this has it written as diphosphate, which over here is pyrophosphate. Those are just the same thing. So I actually have the structure here of the mevalinate pyrophosphate decarboxylase. This is in the program called PyMol, and you get these structures off of the Protein Databank website. And I have the Protein Databank numbers or codes for these in the description down below. So we see this this forms this dimer here, and we can zoom in here on on this the substrate here, which is the mevalinate pyrophosphate. You can see here, and it shows it in 2 different conformations. That's why we kind of see 2 molecules here superimposed on each other. But you can see these orange parts here are the phosphate, so you can see this diphosphate or pyrophosphate. Then I have the carbons in yellow over here, and we can see this is the mevalinate here. We can see, if we move up here, this red is the oxygen on the hydroxyl group, which is what is going to bind to that phosphate. Then we can see down here these 2 oxygens are, this is the carboxylic acid here that will be removed as carbon dioxide. Then we can see, so actually this shows in one of the conformations, it's so close that it's that Pymol thinks that this serine here is bound to this pyrophosphate here. It's almost certainly not actually like that. It's just that this serine is so close to this that Pymol thinks that that must be a bond. And so we can see up here, we can see this arginine which is positively charged, is forming this ionic bond with the negatively charged carboxylate on here. So this aspartate here is probably this aspartate in the mechanism here, which is holding this hydroxyl group in position here. And so we can see that in the structure. Yeah, and then there's this asparagine here, or these two asparagine's, this one right here and this one right here, which, like I said, are both positively charged. So those are stabilizing the negatively charged pyrophosphate or diphosphate here on the mevalinate pyrophosphate. So we can see that how the active site is sort of holding this substrate inside itself. The residues inside the active site accommodate this in the active site here, and so that's the structure of the mevalinate pyrophosphate decarboxylase. And you can see here we have the substrate in the other protein in this dimer here. And so that is the structure and active site of the mevalinate pyrophosphate decarboxylase enzyme. And so if we go back here now, if we want to talk about this MEP pathway, which is actually is a more recently discovered pathway for generating these species here, which are precursors to the vitamin A. And so I'll go through those in a little bit more detail. So if I go over here, we have this MEP pathway, and you'll notice again that that there isn't a fully agreed upon name for these enzymes. So I have them circled in the same color here. So this is DXR, but it's also called IspC and IspD, IspFG, so on. This one over here doesn't have those ISP names, but I have them circled the same way, and I have the mechanisms here circled in the same color. So if we look at this DXR, which is IspC, which is also called 1-deoxy-D-xylulose 5-phosphate reductoisomerase, which is a step in this MEP pathway. I'm not going to go through every enzyme in painstaking detail. Once we get to things like glycolysis and the Krebs cycle and stuff in later on videos, I will probably go through every enzyme in more painstaking detail, but I'm just going to look at a few sample enzymes here. And so first we can look at this IspC. And so in a paper which is linked to in the description down below, they actually looked at the kinetic parameters of this enzyme with the DXP, which is the deoxyxylulose phosphate. So that's what DXP is, which is the substrate. But then they also looked at it with this substrate, which is the same as the DXP, except instead of methyl groups, they have C with fluorine on them, with different amounts of fluorine. And so this is looking at the kinetic parameters based on these substitutions, these fluorine substitutions. And we can see that the K-cat is sort of in the same ballpark. It actually seems to be a little bit faster with the one that's substituted, but if we look at the Km, the smaller number, which is the more tightly binding, so the actual substrate is more tightly binding than the substituted one. And so if we look at our efficiency, the Kcat over Km, the efficiency for the true substrate is higher than it is for the substituted substrate. And so we can look over here at this this mechanism, or at least the reaction here. And so what we're doing is we are turning this here, this acetyl group, into an aldehyde group here. And then we are turning that aldehyde group into a hydroxyl group. And so I put this here because when I show the structure here in a few minutes, it does not have this bound to it. It actually has this bound to it, which is an inhibitor, which is why I drew this inhibitor right here. And so we can see how this might fit into the active site, because we have this aldehyde group here which mimics this, and we have this pyrophosphate group here which mimics the pyrophosphate. And so that's how this can bind, and yet it will not react, and therefore by occupying the active site, it's inhibiting it. And so if we look at the structure here, what we see is we have this this here, which is a manganese ion, and so we see how that is used. So this is our substrate here in yellow. And we can see how this manganese ion can actually be used for the catalysis. And so down here I have the mechanism. So we see our manganese ion here. And so what happens is this manganese ion can actually complex with these oxygens. So it complexes with this double bound oxygen here and this hydroxyl oxygen here. And that's what we see going on right here. So this is our double bond oxygen and our hydroxyl oxygen. And so that's coordinating with the manganese ion. And so the B pathway here is the true pathway with the actual substrate. And so we see that this actually can actually flip this around here, and then we remove a hydrogen, which is an oxidation, because we see that's a hydrogen with a, kind of hard to see, but that's a negative hydrogen ion there. So that's actually called a hydride. And so by no, we're not removing that, we're actually adding it. So we're adding the hydride, and that's how we're reducing this. So that's why it's called a reductoisomerase, because it's reducing it, and isomerase is just sort of because we're changing positions of atoms in the molecule. And so we end up with the hydroxyl group here and the hydroxyl group here, which is this hydroxyl group and this hydroxyl group. So when we add this phosmidomycin, then we end up with the incorrect stereochemistry here. So we can see down here we have moved our double bond so that it's sort of coming out of the screen, where here it's still sort of pointing in this direction, and that does not put it in the right stereochemistry to produce our product here. And so this phosmidomycin will actually not react inside of our enzyme. And so this, we see we have the phosphate there, which we can see over at the end of it here. We can see our two hydroxyl groups, which on this, one of them is actually coming off of a nitrogen, which we can see is in blue right here. But we can see that it's still coordinating with this manganese ion, the big sort of, I don't know, purple sphere there. That's the manganese ion. We can see the manganese ion is held in here by things like this glutamate here. Down here we have this is an NADPH, which is required for this reduction. I don't have it written on here anywhere, but the NADPH is where the electrons are actually coming from, and NADPH is very similar to nicotinamide adenine dinucleotide. In fact, it's nicotinamide adenine dinucleotide phosphate, and that's because on the adenine down here we just have this extra phosphate bound to the third hydroxyl on our. I think that's. No, that would be the second hydroxyl, but anyway, it's the same. So we can see our adenine over here, and then if we move down this molecule, we see our nicotinamide here, and it's this this nitrogen here where our electrons are going to be donated. So that donation is probably going through this manganese, and then it would end up over here if this were the actual substrate. Then if this were the aldehyde, then it would be donating the hydride ion onto that and turning it from this aldehyde into a hydroxyl. And that's where this hydride ion is coming into the reaction to turn this aldehyde into a hydroxyl. And that is a reduction reaction. If we go from an aldehyde, which is bound to a carbon and has an H and then the rest of the molecule. And so when we reduce this, we're basically adding, we're basically taking a hydrogen that has 2 electrons, and so it has a negative charge, and this is coming down and attacking that carbon, which sends those electrons up there, which gives us something like this. Then we have our oxygen with a negative charge, and then that's where we get our H plus, and these electrons will come and take that and generate the so we'll have our H, our R, then we'll have this going on, and these electrons here are where the reduction is happening, but we also have this this H plus here. That's why a lot of times when you see NAD, and I'll even say it's the NADP, it's plus. And so what's a lot of times happening is when this is reduced to turn it into the NADPH version, so we call the hydride that's where the electrons are coming from, and that's what reduces this and turns it into, it neutralizes this. But then we also add the H on it um, because this H here is actually, we're talking about this H right here. And so it comes with the hydride and the hydrogen ion, the hydrogen cation here. And so that's how this reduction happens here. And that is the IspC um, enzyme. And so the next one down here is this IspD, or the 4-diphosphositinol 2C methyl erythritol synthetase. And so this is going to, so we went from our pyruvate and glyceraldehyde 3-phosphate. We formed our we formed our 1-deoxydesylulose 5 phosphate, or DXP here. We went through this this reductoisomerase reaction, and now we are focused here on this reaction. So this is called IspD. It's called this long thing here. It's sometimes called CDP-MB synthetase. Sometimes it's called YGBP. And so all of this is one of the things that can really complicate biochemistry, is having all these different names for things. So a lot of times they'll give something a name like this when we're talking about maybe the yeast version of the enzyme, and then they'll call it this when we're talking about like the animal version, or and so it can get confusing. I won't get too bogged down in that. I'll just try and tell you all of the different names that that you might encounter if you're trying to study this on your own. And so for this, basically all we're doing is we are taking our MEP here and just adding this this cysteine, or not cysteine, this cytosine, that's what it's called. Sorry, brain fart. The cytosine. So this is the cytosine, the same cytosine you would find in DNA. And so we're taking cytosine triphosphate and we are adding it onto here. And we see that cytosine triphosphate plus this, which has a phosphate, is 4 phosphates. And so we are losing this, which is the pyrophosphate. And so we can look at the structure here real quick, and we see here that we have this magnesium. I pointed that out here in the mechanism. So we see we have this magnesium here in magenta, and what we have right here is our cytosine diphosphate here, and then that diphosphate, these two orange things are the phosphates, and then that is bound down here to our MEP. So this is our MEP bound through two phosphates to our cytosine here, and we can see that and this is quite common, is for this magnesium to coordinate, because these phosphates are very negative. So we have this positive magnesium here, which coordinates with these with this diphosphate here. And I have in cyan the residues, the amino acids that are in the active site here, holding these species in the active site. We see over here that we actually have the nitrogens in our peptide bonds from this phenylalanine coordinating with this magnesium to hold it into place there. And we also see that we have this arginine here, which is getting kind of close to these these phosphates here. So that's stabilizing that in there. We can look over here and it might be too far away, but there might be some pi-pi stacking going on with this phenylalanine here. And so the nitrogens on our cytosine are coordinating with the hydroxyl on this serine here, and possibly on this peptide bond on this aspartate here. So this double bond oxygen are hydrogen bond acceptors, and then this nitrogen here would be hydrogen bonding to those. And so we can see how this is all being held inside the active site of this enzyme here. So if we go back here, we can look at our reaction mechanism. So we have our MAP species here, and we have our cytidine triphosphate. And so what we actually see is this oxygen on our MAP is actually attacking the phosphate that's closest, that's most proximal to the cytosine or cytidine, and then that removes these phosphates here. And so we go down here and we see, we can just see the electron pushing going on, where when this attacks here, it causes the electrons to move up on this oxygen, but then they come back down, and it breaks this bond here. And so our pyrophosphate or diphosphate, whichever you prefer calling it, comes off here, and now we have our MAP and this pyrophosphate or diphosphate here bound to our cytosine or cytidine. And so that is the mechanism of this enzyme. So now, so we're skipping this step because this is just basically a similar thing where we're just adding a phosphate onto it, and we'll go down to this one here, MDS, which is also called IspF, or 2C methyl D erythritol 2,4 cyclodiphosphate synthase. And so this is an interesting one because we are actually the phosphate that we just added in the previous step here, we are actually taking that, and that the oxygen on that is actually attacking this phosphate, which will kick off the cytosine. So we'll retain one of the phosphates from it, but it kicks off the cytosine as CMP, and it actually cyclizes this. And so we can see this is the IspF here, and that forms this this trimeric. It's called a homotrimeric because they're each of them are all the same. And then we can see, so what we actually see here is the CMP. We don't actually see in this structure the cyclized version of the MEP, and we don't see the reactant here. So the ion that we actually see in here is the zinc ion. So we don't actually see, so we don't actually see the magnesium ion. But what we see in here is the cytosine. Probably sort of in this area here is where we would have the MEP, and that's where it would cyclize. And interestingly enough, we actually see this Geranylgeranyl pyrophosphate, which is from a later reaction. And we see this binding in this part here where this homotrimeric complex comes together. And what this actually is from the regulatory aspect of this biosynthetic pathway. So a product from a later step in the pathway is coming and binding to this, and that would actually be inhibiting this enzyme. And so if we start getting too much product in a later step here, it'll start binding to this enzyme and inhibiting its enzymatic activity. And so so we can actually see where this product of a later step in the biosynthetic pathway is binding. And you can see they have modeled it in 3 different ways that it could bind in here. And so they probably saw electron density for all 3 different conformations of this Geranylgeranyl pyrophosphate. So you can see the 2 orange phosphorus atoms here in the pyrophosphate. And so that's kind of interesting that you can see where this allosteric inhibition is going on in this IspF protein here. So yeah, like I said, the mechanism is just this cyclizing of the protein, or of the substrate rather. And then so if we go down to the next one, so we will be using, it says these 2 reduced ferredoxin. And so I tried to get the structure for this protein in PyMol, and I could get the structure, but it kept modeling the this ferredoxin, which is this iron sulfur complex, into the wrong place in the model. And so I actually have the paper that it's from here. So the structural insights into a key keratinogenesis related enzyme. Wait, no, so this is a paper for a different enzyme. This is for one from a different, I think for this one I just, yeah, so I have the images. So this is the paper I was thinking of, and so I actually have the images here, and so this is what the enzyme should look like. And so we see we have this cyclic, this Me pyrophosphate, and the C for cyclic, and so we're binding to our IspG, which is going to decyclize it and add this this double bond into it. And so this just shows that when we bond, when this reactant enters our enzyme, then it goes from this open conformation to the closed conformation. And you can see down here this yellow and orange little sort of cubic thing. That is our ferredoxin, which, like I said, I think in the structure I was looking at, it kept trying to model it in the open area where it should be in the in the closed. So we can see we have our substrate bound to that ferredoxin. Then we can look at this, and so this cubic thing here is our ferredoxin, and this is our cyclic substrate here. And we can see as we go from this to this, we are actually we're forming this this radical. So a radical is an atom that has a single. So if we had a carbon like this, and it's bound to 3 other things, instead of being absent a lone pair and being plus, or instead of having the lone pair and being minus, a radical is something that has a single electron and is neutral. But these single electrons really want to pair with something, and so these are highly reactive, and so this will try and pair with something else. And when you're talking about radicals, you always use these half arrows rather than if we had this, we would use a full arrow. And so we end up with this radical intermediate here, uh, and then we go down here, and the radical becomes an anion, and so that electron finds a pair, and then we detach from this ferredoxin group, and that we have our open product here. And so I actually have, so I just had this image here as well. This is the proposed mechanism here. And so we start here at A with our ferredoxin here, and it's coordinated with this glutamate side chain here. And then when our MECPP comes in, our cyclic Me pyrophosphate comes in, this oxygen displaces the glutamate and coordinates with the iron in this ferredoxin here. And then we see that this can form this species here. And so so the electrons seem to move about such that we form this positive here. And then we end up with, we can see this orbital here that has this single electron. That is the radical intermediate there. And so then we can move down here. And so we end up with this carb anion here on the iron. So this carb anion through this mechanism here can sort of move from this carbon to the iron here, and then we are adding electrons, forming this carbo anion here, this negatively charged on our substrate here, and then from this oxygen we actually pull a hydrogen off of this glutamate here, which up here the glutamate obtained the hydrogen in the first place from this, which started as a hydroxyl, but the hydrogen came off onto the glutamate when it coordinated with the iron on here. And then we see that these electrons here collapse down, and then this oxygen here grabs the hydrogen back off of it. And so we recapitulate our hydroxyl here, but we've also now added this double bond here. So this mechanism is a little confusing. A lot of times these redox reactions are a little confusing, and that's why most of the time you end up having a difficult time finding mechanisms like this. You will usually just see it written something like this, where it's just like, oh, we're just adding these electrons, and we end up going from this to this, which now has this double bond here. But yeah, this is the mechanism. I kind of stumbled my way through it, as you can probably tell. But yeah, this is just a redox reaction using this iron on the ferredoxin, and that's how we go from this cyclic species to this HMB PP species. And so that is the last one that I was talking about in this MEP pathway. And so from there we actually split it off into two different species, and those will be our isopentenyl pyrophosphate and our dimethylallyl pyrophosphate, which can turn into each other through this isomerase enzyme here. And that's just moving this double bond from here to here, so that's the difference between those two. And so just to kind of get our bearings here again, so I was taking us through this MEP pathway, which is one pathway in addition to the mevalinate pathway, which, as I said before, is also involved in cholesterol biosynthesis. And so both of these pathways, we get to our dimethylallyl pyrophosphate and our isopentenyl pyrophosphate. And this shows that from here we can use these intermediates to go off and form a number of other things. Then we can use another enzyme to form this Geranylgeranyl pyrophosphate, which that can go off and be turned into other things. And then we can also form it into this farnesyl pyrophosphate, and that can go off and form these farnesines. So basically what we were trying to do through these pathways, though, so far was form this dimethylallyl pyrophosphate and this isopentenyl pyrophosphate. And then we can move on to this next step where now we have formed our isopentenyl pyrophosphate and dimethylallyl pyrophosphate. And so then we can just start adding these isopentenyl pyrophosphates on to form this Geranylgeranyl pyrophosphate, and then we can add another one on to form this farnesyl pyrophosphate. Then we can add yet another one on to form this Geranylgeranyl pyrophosphate. And so the steps on these are all similar, and that's why I only am highlighting a single enzyme in this step, and that is this Geranylgeranyl pyrophosphate. And so we can see here, this is the enzyme for it, and just looking at the residues in the active site, we can look at it here, and then this is just the Geranylgeranyl pyrophosphate. Over here I actually have the Geranylgeranyl pyrophosphate and the farnesyl pyrophosphate, which is this enzyme here and this enzyme here. So I have those aligned over top of each other. And so we can see that there are a lot of structural similarities. The farnesyl pyrophosphate has maybe this alpha helix here that's different. They're a little bit displaced from each other, but you can see there is a lot of structural similarity. If we look at this multiple sequence alignment here, we can actually see that there's quite a bit of sequence similarity, at least in certain places here. So where you see that there is green here, that tells us that there is a lot of similarity. So these are all Geranylgeranyl pyrophosphate from different species. I'm mostly concerned about where these red arrows are pointing, which are the Geranylgeranyl pyrophosphate for humans and the phenyl pyrophosphate for humans. And we see in green here we have these things like these conserved histidines in the different species, but then we have this Q here, which I think is the glutamine, I believe. And so then over here we can see we have these D's and these isoleucines. So the D's are the aspartates, the isoleucines. So we have this conserved region here. I'm not going to go through all of these, but you can also see in places where it's not conserved through the phenyl pyrophosphate. So down here we have a tyrosine instead of a leucine. So the tyrosine is still a hydrophobic enzyme, or a hydrophobic residue rather. So we're not changing the sort of the polarity in that place right there. We have another place where we have these conserved aspartates. We have this conserved phenylalanine right here. And so, yeah. And one thing to notice here that's kind of interesting is uh, there are um, quite a few of these amino acids that are not conserved, and yet our structure is still quite conserved. So that's something you find in proteins quite often is that if you have even something like just a 30% identity between two proteins, this can actually be considered quite high. And you will also see quite often when you're looking at multiple sequence alignments is a similarity rating, uh, sim, simil, similarity, uh, similarity. There we go. There should be an I. And so a lot of times you'll see similarity, and that might be something on the order of like 80%. And that's sort of what I was talking about right here, uh, where we're changing the amino acid, but we're not changing sort of the chemical properties of it by that much. And that's how we get a similarity rating like this. Um, and so, uh, so you see that right here too, uh, and these these yellow regions seem to be showing where there's a lot of similarity. And so just by maintaining similarity, you can oftentimes maintain this structure. And so down here, I have the mechanism, the enzyme mechanism. Um, and so what we see is the pyrophosphate is coming off of our farnesyl pyrophosphate, and that gives us this positive charge right here on this carbon. And so the electrons from this double bond will actually attack this this carbocation here, this positively charged carbon here. And so that forms a bond between this carbon here and this carbon here. And so that would be this bond here. But then we end up with this carbocation here. And then what we see is we actually have this hydride shift. And so it's actually the hydrogen with its lone pair in the negative charge is sort of shifting down this bond here to to uh, to neutralize this carbocation here, and then we are forming a double bond right here, and so that's where this double bond comes from. And we can look at it over here that this pyrophosphate is coordinated with magnesium. This is very common for these phosphates to be neutralized by magnesium. We see that that our pyrophosphate is coming off, which is forming this carbocation here. Our phosphate is pulling this hydrogen off, or at least that's what it shows on this mechanism here, which is forming the double bond here and causing these electrons to go over here, and then we are forming our. So this is actually the mechanism for the Geranylgeranyl pyrophosphate. But like I said, the mechanism for the Geranylgeranyl pyrophosphate, the phenyl pyrophosphate, and the Geranylgeranyl pyrophosphate are all very similar, and we can see that. We can see why that would be just by knowing that the structure of the protein is very similar here. And so I actually have the Geranylgeranyl pyrophosphate here in pymol, and we can zoom in down here. We can see two of our magnesiums here as these sort of gray spheres here, neutralizing this pyrophosphate, the phosphorus being in orange. And then we have our Geranylgeranyl right here, which is our hydrocarbon chain with our methyl groups coming off of it. And we will have double bonds going on in there. And I'm showing the residues here in the active site. And so we can see things like this phenylalanine here, this leucine here. I think that's that's a leucine. Yeah, that's a leucine. And so we can see that these. So I've actually highlighted in purple the residues that are pointing into the next enzyme over in the dimer here. Um, and so we can, what we want to notice here is we have a lot of these, or no, that's in a different enzyme I'm thinking of. These phenylalanines here are actually from the farnesyl pyrophosphate, whereas in the Geranylgeranyl pyrophosphate we instead have these species here, which this is a serine. This should be an oxygen up here, and so those are residues that are different between the farnesyl pyrophosphate and the Geranylgeranyl pyrophosphate. And the paper I got this from says that that these two phenylalanines in the farnesyl pyrophosphate are blocking the active site from allowing longer chains to get in. And that's why down here this farnesyl pyrophosphate, which has this shorter chain than the Geranylgeranyl pyrophosphate, why it would be selective for the farnesyl pyrophosphate. And so, like I was saying before, though, the thing we can notice here is we have a lot of these things like this phenylalanine and this leucine in the active site, and so those are hydrophobic amino acids, and they are accommodating this hydrophobic chain from the Geranylgeranyl pyrophosphate. We have another leucine, or no, this is an alanine up here. And so, yeah, we can see how the two magnesiums down here are accommodating that pyrophosphate, as well as things like this arginine here. We can see that these aspartates and this serine are coordinating with the magnesiums to hold them in place. So this double bond oxygen here is close enough that it would be forming an ionic bond with so this would likely be forming an ionic bond with this arginine here, holding the arginine in place. Well, no, this would be negatively charged, and it would be coordinating with this positively charged magnesium here, and same with this aspartate right here. You can see is very close to these magnesiums, and so that would be coordinating with those magnesiums down there. Yeah, and so we have a lysine right here, which seems like it's possibly coordinating with the pyrophosphate. So anyway, that is the Geranylgeranyl pyrophosphate. We can see that it's forming this dimer here. You can kind of see the magnesiums and stuff in the other protein in this complex. And so yeah, that's the Geranylgeranyl pyrophosphate. So that brings us to we get our Geranylgeranyl pyrophosphate So this is just so that brings us to this step right here. And so now we want to take our Geranylgeranyl pyrophosphate and turn it into this species, which is phytoene, which is a precursor for carotenoids. And so these steps right here, the phytoene synthase, the phytoene desaturase, these are things not found in humans. And so that is why we cannot produce beta-carotene here. And so what's happening is we're taking our Geranylgeranyl pyrophosphate, or we're taking two of them, and we're combining them into this phytoene here, which has this cis double bond here. Then phytoene desaturase is turning this cis double bond into a trans double bond here. And then there will be these cyclases, which just, well, they cyclize the ends of this lycopene here, and so it's forming this six-member ring. And so that would be this 2, 3, 4, 5, 6 carbons. So these carbons here are ending up in this ring right here in our beta-carotene. And if you know anything about genetically modified organisms or GMO food, you might have heard of something called golden rice. And golden rice is adding these genes into rice, so that rice, which does not have the phytoene synthase and phytoene desaturase naturally in the wild, it cannot turn its Geranylgeranyl pyrophosphate or diphosphate. Like I said, those are used kind of interchangeably. It cannot turn that into the lycopene and then cyclize that into beta-carotene. And so the GMO golden rice adds these genes into the rice so that the rice can generate beta-carotene, and that's to help people in developing countries get enough vitamin A in their diet. And so that's what golden rice is it's adding these steps to the rice. So the phytoene synthase, and this is the one I was thinking about earlier when I went to that paper, is one that I wasn't able to find a structure for. So this paper is actually doing a structural prediction. It's actually using structural prediction software to try and guess what the I mean, it's not just guessing, it's using various algorithms and things known about protein folding and all that kind of stuff to try and develop or predict a structure for the phytoene synthase. And this is a 2015 paper. And so in this paper, so right here, I will rotate this so you can see it better. They are trying to predict secondary structures. And so we see where they have these magenta little cylinders here that is trying to predict alpha helices. And these bars up here are their prediction confidence. And so you see they have more confidence in these structured areas, where when it's unstructured, it's harder to have good confidence because those are often loop areas that are sort of more unstructured or floppy, to be very scientific about it. And so you can see these arrows are where they're predicting some beta sheet. So they're predicting a lot of alpha helices in this, and these are the protein structures that they are predicting. And you can see here they have their Ramachandran plots, and all the black dots are where their dihedral angles are ending up in their Ramachandran plots. Uh, you can see there's a few of these red guys out here outside of the allowed regions. Um, that's not uncommon because sometimes just because of things like side chain clashes that you need to twist the dihedrals into angles that are outside of the quote unquote allowed range. But you can also see that they have their proline allowed ones in here, and their glycine allowed. So proline is the one that has that cycle that goes from its alpha carbon to its nitrogen, and so they have the weird sort of bond angles, the weird sort of dihedral angles going on. And the glycine, which does not have a side chain, is very flexible. So those have kind of a wider range of allowed dihedral angles. And so these are the protein structures that they predicted. These were not obtained from X-ray crystallography like all the other ones were, or cryo-EM, which is fast becoming very common for obtaining structures. So we'll rotate this back around, and so here is their multiple sequence alignment, and you can see for different species here. So the Plasmodium, the Toxoplasma, and the Thermatoga. So those are all just different, basically different bacteria. And so we can see here that the blue seems to represent nonpolar or hydrophobic, and so we can see that there is a lot of conserved hydrophobic residues in it. The yellow is our proline, so there's quite a bit of proline in these. And so, yeah, these sort of red and purple are charged. So you can see that things like this arginine here, the arginine and lysine up here are quite conserved in these three different species. And these are the structural predictions from the three different species. And you can see in the Toxoplasma gondii and the Plasmodium falciparum, they're predicting these loops up here, these unstructured regions in their protein, and so these are motifs that they've found in it. And a motif is basically just a small section of amino acids that have these conserved sort of residues. So you can see here we have the conserved glutamate, and then the conserved hydrophobic, and we have the conserved arginine here. So that's their motif 1, and then they have their motif 2 here, which has the conserved aspartate, and then 2 amino acids, then aspartate. And so these are where they're predicting their motifs to be in the protein. And this is a structural alignment between this phytoene synthase and this OPPS protein, which has a similar mechanism of action. And so you can see that the active sites of this OPPS, which is a known structure, and their PSY, which is the phytoene synthase, which is their predicted structure. You can see that they have a similar mechanism of action, and they overlap quite well in their predicted structure. And here they give the active site residues for the OPP, which has the protein databank code of 1V4E. And then the residues in their predicted structure are down here. And so this is the mechanism of it. So we have our two Geranylgeranyl pyrophosphates here, and we're essentially doing that same thing as we saw before, where we are forming a carbocation by removing this pyrophosphate. The double bond from this is bonding to it. We have this hydride shift here. We are removing this pyrophosphate. We actually have this three member ring here, which 3-member rings are very unstable, so this is not going to exist for very long. So this bond here is shifting up to this carbocation that's being produced by losing this pyrophosphate. And so losing pyrophosphates is very energetically favorable, and so that's why to do this reaction they are using these pyrophosphates, which is removing pyrophosphates is very energetically favorable. And so that's what's driving this reaction. Forming carbocations is not very favorable. So in order to do that, they are removing pyrophosphate to generate these carbocations. And then this bond moves up here. We end up with this 4-member ring. Then we have our NADPH, which is actually reducing this right here, and it causes this bond to move up or this bond to break here and form a double bond here, which then we form our phytoene, or it can be our squalene. And we will see more about squalene in later videos, because this is involved in the production of other vitamins, particularly vitamin D, in fact. But right now we're focusing on this phytoene, which is this species here, which is our 2 Geranylgeranyl pyrophosphates bound together with this cis bond. And then we actually want to turn that cis bond into a trans bond using this desaturase. And that is this mechanism over here. So there are actually 2 different ways this can happen. So archaea, bacteria, and fungi actually use a single enzyme to take this phytoene and turn it into the all-trans lycopene. And so plants, which would be things like your carrots and your spinach and the things that we usually get vitamin A from, and cyanobacteria, which are photosynthetic bacteria, actually go through this more convoluted process here. And so they actually turn this into a cis double bond with this PDS enzyme, and then they do the same to this sort of symmetric double bond, turn that to cis, and then they actually use a different enzyme to then change all of those into trans double bonds. So the archaea, bacteria, and fungi actually do that all in a, well, they don't do any of these intermediate steps. They just take this 15-15 prime cis double bond and turn it into a trans double bond. And so the mechanism for that, or at least proposed mechanisms, so one of the things we need for this, we can see here we have the FAD, which is flavin adenine dinucleotide, which is it's similar to NADPH, which was the nicotinamide adenine dinucleotide, and that's doing the redox reaction, and it's passing those electrons onto the plastoquinone, which is this up here. And so we can see when we reduce it, we take this ketone here and turn it into hydroxyl. And then we reduce it further, taking this oxygen here into another hydroxyl, and you can also reoxidize it back to this double bond form. And you can see that from the FAD to the FAD is taking electrons from this, and which is reducing the FAD, which then passes the electrons onto the plastoquinone, turning it from this into this, and then the plastoquinone actually passes that on to oxygen here, which would then turn that into water. And so this is kind of this convoluted process here. This is the redox of, so this is the FAD, the full molecule here, the flavin adenine dinucleotide. And so we're going to focus on the flavin part of it up here, and that's what we have going on up here. So this is the redox of flavin adenine dinucleotide. And so when we reduce it, we take this nitrogen here, and we add a hydrogen onto here, and we take electrons with it. So we are adding a hydrogen cation, but also this hydride ion here, and these nitrogens here are now hydrogen or nitrogens with hydrogens rather than double bonds, with the double bond moving here rather than to the nitrogens here. And then we can, when we remove one of the electrons, we end up with this radical here, but then we will quickly remove that electron to the plastoquinone along with a hydrogen and get back to our oxidized form of the flavin adenine dinucleotide. So over here are some proposed mechanisms for this. And so I don't want to go into these too much. Essentially what we're trying to do though is turn a trans double bond. We want to get rid of the double bond, which cannot rotate around the bond. It's held in its position. We want to sort of get rid of that double bond so that we can then rotate around that bond, and then we can, so we've rotated the CH3 so that we can put it from the trans into the cis position here, or the other way around. So it's just a way of changing from cis to trans. This reaction could go in either direction. This phytoene desaturase could go in either direction, but here we are trying to turn a cis into a trans, and so we might go from this turning our cis here. We sort of remove the double bond character. We oxidize this, which was that step over here. So we're oxidizing it. Our FAD is removing electrons from it. That's oxidation. So we're removing electrons, which turns this double bond into a single bond with this carbocation here. And then we can swivel, we can rotate around this bond here, and that allows us to go from the CH3 down here to the CH3 trans here, going from the cis double bond to the trans double bond. And so I do actually have the phytoene desaturase here. We can see in green right here, we can see our FAD, our flavin adenine dinucleotide. And right here, this sort of three-member ring right here is our flavin part right here. And so we can see the flavin is near where our substrate would be. This this molecule here, which is next to the flavin, is not actually our substrate. It's actually an inhibitor. It is called norfluorazane. Norfluorazane. I think I might have. No, I didn't. Uh, I didn't put a molecular structure in here, but we can see the molecular structure in this. It has these two rings on it, and so that goes into the active site and where our substrate would be, which is this phytoene synthase, and it's inhibiting this enzyme here. So you can see how that goes close to this FAD, to the flavin here, which if we had our substrate in here, this would be close enough that it could be pulling electrons off of our substrate. And so we can see, and so I changed the color of this alpha helix up here because I thought it was kind of funny. The paper that I got this structure from, which is in the links in the description, calls this the hot dog region up here. So I think they're thinking of this alpha helix like a hot dog, with these beta sheets being like the hot dog bun. But anyway, I thought that was kind of funny. And so this exists as this homotetramer. So there are 4 of these sort of bound together, and this protein actually sits on top of a phospholipid bilayer membrane. So there would actually be membrane down here that this is sitting on top of, and so I've highlighted here in gray the amino acids that form the interface between the different proteins in this homotetramer. I have in cyan here the active site residues, so you can see the cyan here where the substrate would be sitting inside of this active site here, and what you can see is a lot of these are hydrophobic amino acids. So things like this, we have a leucine here, we have another leucine over here, we have a tyrosine right here, which it has this on here, but its ring kind of dominates it and makes it hydrophobic. We have a couple phenylalanines down there. We have a methionine down here, which would be hydrophobic. And so those hydrophobic residues are accommodating this large hydrophobic chain inside of the active site. I did not get the structure for the lycopene beta cyclase. We'll actually look at cyclases a bit more when I talk about vitamin E in the next video. So I won't focus on that here, but we do cyclize on both ends of these carbons, and turn it into these 6-member rings. And that's our beta-carotene, which the beta-carotene we then cleave into 2 these species called Retinal, which this is then reduced to this aldehyde here is reduced to form this hydroxyl group here, and that gives us our vitamin A. And so this cleavage here is performed by what's called the 15-15 dioxygenase. 15-15 prime, because we're counting from here up to here on one side, then here up to here on the other side. We get to the 15th carbon on both. And to distinguish them, we call one 15 prime and one 15. And so using this 15-15 prime dioxygenase with oxygen, uh, we form this four-member peroxide. So an oxygen single bond to an oxygen is called a peroxide. So you will also see this sometimes called 15 prime peroxidase. Uh, and so we form that, which then allows the breakage of these bonds. And from one beta-carotene, we form two retinols, which can then both be used to form two retinols or vitamin A. And so I have, so this is not the mechanism of the 15 dioxygenase, but it's the mechanism of a dioxygenase. And this is actually the dioxygenase for resveratrol, which is a molecule I will talk about in a later video. But this is the same mechanism in that we are breaking this double bond using this intermediate of the oxygen peroxidase four-member ring, and then forming this cleaving it here to form these two aldehyde species. And this, you can see the energy diagram here. We are moving from a higher energy state here to a lower energy state here, but we have to go through these higher energy transition states along the way, and that's what and the enzyme is helping to facilitate this. I actually have a mechanism over here that's a little bit more in depth, and so we can see that our oxygen, just like in hemoglobin, oxygen is binding to this hydrogen here, is coordinating with this hydrogen, and this coordination becomes a bond. So this bond here sort of sweeps up there and becomes a bond to the iron up here. Then we end up with this free radical, and you can see these free radical dispersion, I guess. Dispersion, not a good word. That has that actually has a meaning in quantum mechanics. The electrons from this double bond. So one of them is going to here, one of them is going here, and one of these double bonds is going here, one up here. And so we end up with these radical electrons here, which will then form a bond, because these radical electrons will want to come together and pair up to form a bond. And so we have the oxygen bound to the iron, and then to the molecule here, through some more radical electron movement here. So we see we ended up with a radical at the end here, and with this double bond. One of the electrons from the double bond will go up to here, one down to here, so we reform our aromatic ring, while also generating this oxygen, this peroxide species here, this peroxide. This would actually be a four-member ring, like we saw in our previous mechanism here. And then because we still have this radical here, we're going to do some more radical electron movements around to recapitulate our iron in its normal state, just like we need in a catalyst. The catalyst speeds up the reaction but does not get used, so we need our iron in this 2+, just like we had up here at the beginning, and we now have these two aldehyde species here. And like I said, this is on this still bean. Resveratrol is a type of still bean, and it has this trans double bond here which you will find in resveratrol and other still beans, and we go from there. We cleave that bond and form these two aldehyde species. And that's what's going on with this 15,15 dioxygenase. And so I actually have that here, so we can see our beta-carotene right here, or at least a beta-carotene analog. And we see in red here, this is our iron ion species, and this is sort of notably not in a heme group. We'll run into iron in heme groups later on. Heme from the root for hemoglobin, and it is also not in that ferredoxin, that iron sulfur molecule that we saw earlier. This iron is by itself, and you can see all these histidines around it, these rings here that are coordinating with our iron to keep it in place. So these histidines are sort of serving the purpose of a heme in this instance, and just holding our iron ion inside the active site of our enzyme here. And then this up here, which is our beta-carotene analog, at least. It looks very similar to beta-carotene. We can see it has a 6-member ring here, but it seems to be missing a 6-member ring at the other end, and it seems to have trans double bonds in it. But this would be the double bond that that this iron is attaching oxygens to in order to break this bond here and turn it into two different things. And so we can see the structure here has quite a bit of beta sheet in it for our 15,15 dioxygenase species. And so that, yeah, that's breaking that bond on our beta-carotene. And so this is something that humans can do. And so that's why beta-carotene is a source of vitamin A, because humans can go through these enzymatic processes and turn beta-carotene into vitamin A. We just cannot do these right here. We cannot turn geranylgeranyl, pyrophosphate, or diphosphate, whatever you want to call it. We cannot turn that into beta-carotene. So we need to get beta-carotene from our diet, or we can get it in one of these forms. I think there are some animals, particularly predator animals, if I remember correctly, that do not do these. So animals that are strictly predators, I think they have to get their vitamin A as vitamin A, but humans can acquire it as beta-carotene. And so that is the biosynthetic pathway that brings us from sugar. So we go, well, we don't. Organisms of some kind do, particularly plants, can use sugar, go through this MEP or this mevalinate pathway to form the geranylpyrophosphate, the phenyl pyrophosphate, which that is then used to generate this geranylgeranyl pyrophosphate. Then that geranylgeranyl pyrophosphate, which we have right here, is through this phytoene synthetase and phytoene desaturase and lycopene beta cyclase. So the golden rice GMO enzymes, or the enzymes that are put into rice to make them into GMO golden rice. And that we go from there, from our sugar to our geranylgeranyl pyrophosphate to our beta-carotene, and from beta-carotene to our vitamin A. And so that is the biosynthetic pathway of vitamin A. In the next video, I will go through vitamin E, and later on, and then after that, I'll do vitamin K and vitamin D, which both use a lot of the same things. So a lot of them use these Geranylgeranyl pyrophosphate molecules here. For instance, I think I had that somewhere. So yeah, the Geranylgeranyl pyrophosphate. So yeah, if we go down here, the squalene, which we can make from our Geranylgeranyl and our phenyl pyrophosphates, can be used to make squalene. And then squalene is used to make vitamin D. The Geranylgeranyl pyrophosphates are used for different things when we make vitamin E and vitamin K. They do not use the phytoene synthase and so forth, but we will get to that in the next few videos, so I will see you then.